So while we are predicting the genes, there are two famous approaches which we use them in order to predict the genes from some sequences. One of them is we predict them from ab initio approaches, where we just look into some signals present in that particular DNA and predict our genes from there. The other approach is called as comparative approach. In comparative approach, we compare that sequence to some other sequences. We do some sequence alignments or comparisons, and in this way we can align our sequence to some known gene sequences, and if similarities are high, we can expect that there is a gene which is present. In ab initio approaches, we detect genes by looking for distinct patterns that help us define a gene. These ab initio approaches try to kind of overestimate the gene numbers. So any segment that looks like those patterns, it might give him a call as a gene, but that may not be true. So this kind of issue is we call it as overestimation. Whereas comparative approaches, they look for genes by comparing segments of sequences with the sequences of known genes. But this has an issue of underfitting, or we can say the issue of underestimating the genes, since it is limited to recognize the genes by comparing to the known sequences only. So it's hard to find what is novel or what is new. Okay, so we see how the gene structure looks like and what patterns we can go and find it in order to give a gene call. So a gene in case of eukaryotes looks like this. So we have a promoter sequence. It's the sequence where we have some specific patterns where some proteins, we call them as DNA binding proteins, they come and attach, and then they help starting this transcriptions. Just recall guys, transcription is the formation of RNA from DNA, whereas translation is formation of protein from that RNA. So here is the promoter in this gene, and then this actually this transcription starts from somewhere here. We call it as transcription start site, that is TSS. Then there is a region we call it as 5' UTR, untranslated region. So that part goes into the RNA, but it is not translated. So protein is not formed from this region, but still since this transcription starts from here, that's why it is TSS. Translation starts from this start codon, so that is ATG. You know that these amino acids, they are incorporated into the proteins while reading these nucleotides in the shape of these triplets. We call them as triplet codons. So ATG encodes for methionines, and that is true for most of the polypeptides. There might have some few exceptions, but this is kind of a universal thing. Then we have exon 1. So you see in eukaryote, this ATG or start point is in exon 1. In between there is an intron, and then we have exon 2, and then we have those stop codons which are here, TEA, TAG, TGA. So this is how the translation later on, it will stop with these codons. But as far as transcription is concerned, it stops here. So this stop sign for transcription. So messenger RNA will be formed from these regions, and obviously these UTRs are later on, they are there, but they are not there in the final product. So after we get this pre-messenger RNA, we get the final messenger RNA where introns are chopped out, and we get exon 1 and exon 2 joined together. And this is the start codon, and these are the stop codons. So in order to do the promoter prediction, we want to find some patterns in the promoters. In order to get a gene, you need a promoter. You can predict this TSS site. You can find the presence of ATG. You can look into those stop codons, and all these things, all these patterns, they are important in gene finding. The place where we have this start codon until stop codon can also be called as ORF, open reading frame. So that is the place from where we will get the proteins. And these are predicted ones. Will we be getting the proteins or not? That depends. So most of the time when we are doing those ORF findings, we have so many ORFs. Some of them are right, or some of them may be some false positives. Coding sequence That is another term which we will use later on. So coding sequence is the sequence that actually goes into your protein. So that is exon 1, 2, start, stop codon. So that whole chunk or the fragment is called as coding sequence. As far as this gene finding is concerned, there are some other issues. So we know that in human, there are 2 to 3% DNA encodes for functional genes, and genes are interspersed among long stretches of non-coding regions. So very few part has genes, and we have so much non-coding region. Earlier we used the term junk, but still we are getting some interesting patterns out of it. So don't use the word junk, but still in the literature you would find the word junk DNA. Then in case of the human DNA, our eukaryotes, we observed there are large number of repeats, so up to 40%. And then there is the great variation in the gene length. So some genes are long and some are shorter. So you cannot come up with an exact number so as far as the length is concerned. We also have to distinguish between pseudogenes and some genes which are arrived due to this gene duplications. And there are some other issues that make the situation complicated, like exon intron structures, non-consensus splice sites. We'll talk about them when we talk about splice sites. And where is the first exon? That is also an important question. Sometimes we try to capture these genes while taking those cDNAs. We take the messenger RNA and get their DNA copy out of it. And since that part is what is expressed, or that part is which is coming from the genes, so we can get back to genes if we use that cDNA. But most of the time we see the situations where we have issues with cDNAs. Alternative splicing and chimeric genes and alternative promoters, they might also add complexity. So in this section, what we have seen here is that there are two approaches with which we can do this gene prediction. One of them is ab initio, if you start from scratch, or you just try to get the patterns directly from that sequence. So if you are comparing with some known genes, that approach is called as comparative, and gene finding is not an easy task.